Hello, everyone. It's 11, but I'll give a couple more minutes before I begin the workshop. Meanwhile, my colleague Esteban would be dropping the useful links in the resources. If and you can also see it in the screen, I guess, on the slides.
Yes, giving up the others.
Okay, it's 11:02, and I will start the workshop. Welcome, everyone. Thanks for joining the workshop to learn about a tool called Morpheus and how you can use it to explore the information that you get from microscopy images. I'm Suganya Sivagurunathan, a postdoctoral associate at Betsamani's lab in Broad Institute of MIT and Harvard. Today with me, Esteban, Lei, and Satavisha, who are my colleagues, will be joining, helping you during the hands-on exercise, which you will be doing during the later part of the workshop today. Before I jump into the Morpheus tool, I would like to ask a favor from you all. I would appreciate if you all could contribute to the survey. That is a joint survey conducted by Center for Open Bioimage Analysis, Bioimage in North America, Royal Microscopical Society, and GloBIOS, the Global Bioimage Analysis Society. And the intention of the survey is to understand the needs of the imaging community, especially in the terms of image analysis. The duration of the survey is not that long. It takes only 5 to 15 minutes. And you can find the link here and also a QR code, which you can scan later to fill up the survey. And the resources are also made available in the resource tab in the Zoom. And these are the links for downloading the materials which we'll be using for the later hands-on exercise part. And the second one is the link to the Morpheus software. And the QR code is again a link to the materials that we will be using later today. And these links will also be available in the resources tab. Yeah, so I have structured the workshop in such a way where I'll be giving you an overview of morphological profiling. Why I'll be talking about morphological profiling is I'll be showing the use of the Morpheus tool, explaining morphological profiling. And then I'll give a demonstration of Morpheus using the various features, how you can explore your microscope information that you obtained from microscope images. And then the third part would be the hands-on exercise. First, I'll jump into the overview of morphological profiling. Unlike old days where we record the microscopy observations by hand drawing all the information that we observe, with the introduction of digital cameras, every other microscopy image that we capture today are nothing but an array of numbers. For example, the image that you can see on the left is each square in the image you can consider as the pixels in your microscopy images. And each of those block of squares has a value. As you can see on the right, it is an array of numbers that you see in the image. So each square block has some value associated with it. This is something so similar in microscopy image as well, because each pixel in your microscopy image contains some values. If it is a fluorescence image, each of it would contain values about the intensity of your fluorescence, so on and so forth. So if you're having an image of 1000 by 1000 pixels, then each of the pixel would have some information or numbers associated with that. If this is the case of just one single plane image. If you're having multiple Z planes, like a 3D, three-dimensional volume image, or if you're having a time-lapse movie, you can imagine how much of data that you get from a single microscopy image from your experiment. But are we using all those data to understand the biology that's going behind? I'll go into details of what we generally do. So, in a typical quantitative microscopy experiment, a biologist comes up with a hypothesis like a particular perturbation or a treatment would alter a certain organelle or protein of interest. So, what we generally do is we tag the protein of interest or an organelle of interest and then give the perturbations or perturb the cells and then image them. Once we have the images, then we look into the images to understand the changes that are going on the protein of interest or the tagged organelle of interest. We measure the features or see what are the changes that are associated with the protein or organelle of interest, and then we come up with biological insights. So if you consider this single cell to be one microscopy image that you get, you have information in the whole image, but what you're using is just the tagged protein of interest and not the whole information. So there's a whole level of information from the microscopic image that we are missing that we are not using. So that's what we are trying to do in image-based profiling experiment. What we do here is we let the cells speak for themselves. What do I mean by that? Rather than tagging a particular protein of interest or organelle of interest, we try to stain all possible organelles in the cells and then perturb the cells. Rather than focusing on a particular organelle or a protein of interest, we now try to see if there are phenotypic changes associated with the particular perturbations. And then if there's a pattern in the perturb samples or the control samples, or if there is a pattern between the perturb samples in their phenotype, then we try to understand, based on the pattern of their phenotypic data, we try to understand if there is a new hidden biology in the microscopy images that you obtain. But how do we do this? In a typical workflow in a lab, what we do is what I'm going to be discussing with you right now. This is an image-based profiling workflow. We also call the image-based profiling as morphological profiling. This is generally done in a high throughput manner where we culture cells from control or healthy patients, or if you are having a certain cell of interest, cell line of interest, then you can culture them in a multiple plate and then give them perturbations. The perturbations can be different compounds of interest or drugs, or if you're interested in studying about a particular gene, so it could also be like genetic overexpression or gene knockdown. So what we do is like we culture all these cells in a multiple plate, give all the perturbations based upon whatever the perturbation of interest is. And then once we have given the perturbations, we fix the cells and stain them with 6 different stains in our case. And these 6 different stains would be labeling 8 different compartments. And the compartments that it would be labeling are nucleus, endoplasmic reticulum, mitochondria, actin, Golgi apparatus, plasma membrane, nucleoli, and cytoplasmic RNA. And all the information of the organelles are captured in different image channels. And once we have all the images, in this case, the objects of interest would be cells. But in your case, if you're interested in studying about an organoid, the objects of interest could be organoids as well, or any objects or anything that you're interested in studying. Once we have all the images, then the next thing what we do is we segment the objects of interest. Since we have cell lines here, we segment the nuclei and the associated cells that are in the particular perturbed cells or the control conditions. Once we have all the objects of interest, the next thing we do is extract the features. What do I mean by features here? Any information that you get about these objects, the segmented objects, be it a nuclei or a cell, any information about these objects, that is what we consider as features. For example, it could be the area of nuclei or the area of the cell, or the intensity information or granularity or texture. We typically use Cell Profiler to extract all the information or the features from these segmented objects. So now we have like a lot of information about the cells, both from the control as well as the perturb conditions. So now, since all of these are in numbers, what we can do is then we can represent these in a heat map, as you can see here. Um, what this heat map represents, each column in this heat map corresponds to a particular perturbation. It will contain the control cells as well as, and the other column might be the information that you got out of the perturb cells. And each of the rows corresponds to the different features that you would have obtained from Cell Profiler or any other image analysis software that you use to extract the features. Once we have this heat map, you can now imagine this heat map something similar to a transcriptomic or proteomic profile where it would correspond to the gene or gene expression or protein expression levels. But here, instead of the gene or protein level expression, what you're looking at are the features that you obtained from the images. So you can call these as morphological profile, similar to what you would call a transcriptomic or proteomic profile. Once you have this heat map, then it is kind of easier to understand the pattern that is going on based on the perturbations. Like with the information that you obtain, you can then cluster to see if there is a pattern in the phenotypic changes that is going on in the perturbations, or how different or dissimilar or similar are the perturbations to the controls. But how do we have obtained the features from Cell Profiler and we have like a CSV or an Excel sheet of all the values, and you can make it a heat map or understand a cluster. But how do we do all these? That's where Morpheus comes in. Morpheus is a web-based software initially developed by a different group at Broad Institute to visualize the gene expression data. So in Morpheus, we use something called similarity matrices to understand the association that is going on in the data, be it your morphological profile or a gene expression profile. But today we'll be focusing on the morphological profile or the image-based profiles. So yeah, this is a free web-based software. As I mentioned, this was designed at a different group in Broad Institute to visualize the gene expression data, and it will accept multiple formats, file formats such as CSV, GCT, text file, and yeah, and it allows matrix visualization. You can also analyze or cluster the data that you have obtained, and you can also filter the features that you obtained, and you can also make charts using this Morpheus software, which I'll be showing in the demonstration in the later part. And the very nice thing about Morpheus is we don't need extensive computational experience because it's a web-based software. There's no installation required. You can directly import the data that you would have obtained from your images into the Morpheus and look at the association that is going and understand the biology. So the applications of the software would be like, again, to examine the correlations between the samples. And if there is a correlation, if you see a positive or negative correlation, you can see what are the features that drive these correlations, and which would then help you to understand the biology that is going on with the microscopy images that you have. So in the next few slides, I will share with you the workflow that we generally do to understand the association that we get from the microscopy images. This is like a normal 4-step workflow, which I'll be going in detail of each of the steps in the following slides. So the first one is the extraction of features. So you would have perturbed the cells, imaged them, and segmented the objects out. So you can extract the features either using Cell Profiler, which is what we typically do, but this can also be done with any image analysis software, which would help you in extracting all the features. Again, by features, I mean the area, texture, or granularity, or any other information that you can get from the segmented objects of the microscopy images. And the second one is average Well, that's what we generally do when we do image-based profiling. What do I mean by average Well, or why do we do that? When you consider a high throughput experiment, like when you have like a 96 well plate like these, I don't know if you see my cursor here, but each well would have different treatments. And each of the well will have a lot of different cells, and some of the cells might be in their cell division stage, and there will be a lot of heterogeneity that is going on in a single well. So we wanted to eliminate the heterogeneity that are associated with a single well so that the later part of the clustering gets easier. And it is also easier to compare if you're doing it in a 96 well, compare 96 wells by 96 wells, than compare a million of cells against each other. And since we are Taking an average per well or per well means this tool can be used to look at major systemic changes and not the subtle ones. Once we have per well means, there are a couple of pre-processing steps before we go into understanding the association. The first one is the normalization. Like any other biological experiment, we also have to do like a normalization here. And I will show why this normalization part is important in the demonstration. And we either do it with the negative controls that is available in the data or to the whole data that we have. Once we have the normalized data, then what we do is a step called feature selection. We would have obtained a lot of features from the images. For example, if you're using Cell Profiler, we obtain thousands of features from a single image. But with having obtained all the features, why do you want to select only a subset? Why not use the entire set of the features that you have extracted from the images? Why do we subset is the next step where we want to associate based on a correlation coefficient. The correlation coefficient would be affected if you have like a redundant set of features. So we don't want to be adding noise in the data when we are understanding the association. So it is important to remove the redundant features when we are looking at the association. So these are the pre-processing steps that we would do before going into the similarity matrices, which again, I'll be showing the normalization and the feature selection details later in the demonstration. So once we have done the pre-processing step, the next one is the Pearson correlation coefficient. Pearson correlation coefficient is a way to represent the similarity where we try to associate, where we measure the strength of the linear relationship between two variables. What do I mean by that? Here in the graph, when you look at it, look at the graph, what this graph shows is we are plotting all the features that we have obtained from one well to other features that we have obtained from other well. For example, here we are comparing the feature, we have plotted the features from well C2 to well B2, and the Pearson correlation coefficient is 0.9. What it means is the similarity between the features from these two different wells is 0.9. A Pearson correlation coefficient of 0.9 means they are highly correlated, whereas the negative values of Pearson correlation means they are anti-correlated. So any values that are around 0 means there is no correlation. So the Pearson correlation coefficient is just a way to start comparing seeing the linear relationship between the two variables. We can do other coefficients as well, but we generally begin with the Pearson correlation coefficient. So based upon the values that you obtain with the Pearson correlation coefficient, you can now say how similar or dissimilar each of the compared wells are. This could be your control wells or the perturb wells. So now we have the information for two different wells, but how do you study it for the whole 96 well plate or a multi-well plate that you would have imaged? That is where the similarity matrix comes in. So similarity matrix is a way to assess the covariance in features between all pairs of columns or rows. What we saw in the previous slide where we just compared the coefficient between two different wells, but we want to do it across all the data that we have. So the image that you see here, like which is more like a heat map, you can see like each of these squares are nothing but the Pearson correlation coefficient of what is being compared. All the columns are what you see here are different wells. Like the different wells corresponds to different treatments in this case, where um, but here I have shown only the DMSO treatment, but you can see these are each square represents the Pearson correlation coefficient, and each well, like the well B2, is compared to a different well. In the first case, it is just a B2, but in other cases, the B2 is compared to the other wells in the experiment. Again, I'll show how we can do this during the demonstration. By having a matrix like this, we'll then be able to associate what are the correlations between the different perturbations that you get from a multi-well plate. Again, I keep saying about the multi-well plate, but this can also be done in for other kind of experiments as well. Having said all this, I would like to point out that there are some caveats when you're looking into the association that you get from image-based profiles or the morphological profile. Since we are getting the features based on the segmented objects, we need to make sure the segmentation is good enough. It is not sufficient for the segmentation to be good enough only in your control cells. We need to make sure the segmentation is good enough across all the perturbations. Because when you look at the association later, whatever the difference that you observe uh, in the like in the similarity matrix should be the result of your differences in the biology and not because of the segmentation differences. So that's why we need to make sure the segmentation is good across all the different conditions in your data. But how do you say when the segmentation is good enough? I have linked a blog post in the resources slide in the slide deck, which you can use as a guideline for saying the segmentation is good enough or not. And the other important thing that you need to take into account is since these are all this is like a starting point to explore the biology that is hidden behind. You shouldn't consider this as like a endpoint because whatever the association you see is like indicating there might be some biological interesting biology that's going behind, but which needs to be confirmed with other experiments as well. And at each point of your experiment or the image-based profiling experiment, you need to make sure that you have an idea of the images that you have captured and also understand the feature information that you're handling, because these are critical. This can be further emphasized by the fact that on the right, you can see there are different plots with different kind of patterns. All of these plots have same mean and standard deviation of the data that they have. But when you plot it, you can see different structures that comes out of it. So that's why it's very important to have an idea of what is the data, what is your data, both at the image stage as well as at the bioinformatics stage. So here I would conclude saying that the morphological profiling is a very powerful way to understand if there is a pattern associated with the cellular phenotype and the different perturbations that you're studying. And you can get these phenotype data using Cell Profiler or any other image analysis software that you can use to extract these features. And with Morpheus, you'll be able to normalize and understand the biological insights that is going on with the microscopy images. And uh, and again, the main advantage of Morpheus is all these can be done without the use of command line. So with this, I would stop here for to know if there are any questions, and then I can go to the demonstration part. Okay, I don't see any questions, so I will jump into the demonstration. I have there should be a link to the Morpheus software in the resources tab, and also uh, you can also I'll show the different features that can be done with the Morpheus, which you can later try during the hands-on exercise. Do you see my Morpheus screen? Okay. So this is the interface of how it generally looks like when you open the Morpheus software. I already have a GCT file that we have obtained from an experiment where the perturbations were different compounds. So I'm going to be using that GCT file to show you how the similarity matrices and other things can be done with the Morpheus tool. So I've just dropped or selected the particular GCT file and I've opened it. So once you open the GCT file, this is how it looks. All the individual rows are the different features that you obtained from Cell Profiler, or if it is a different software, then the rows would correspond to the different features that you obtained. But we don't know what's going on with the columns here. To make it more meaningful, what we can do is we can choose the gear button on top, which is the options. Once you choose that, you have three different tabs that are shown here. One is annotations, color scheme, and display. I would go in annotations. And under annotation, there are two different things that you can choose. One is row annotations and the column. The row annotations correspond to the CP feature name, which is nothing but cell profiler feature name, which we already have the information for the row. What we are missing is the column annotation. So when you click this drop down, you have the option of choosing compound, the compounds that the cells were treated with, and the concentration of the compounds. MOA is nothing but the mechanism of action of the different compounds that the cells were perturbed with. and the details of the plate, and also the wells from where the information were from. I would uncheck the ID because that is not adding much to the much information to us. So once that is done, you can now see that each column corresponds to the information that you have obtained from different wells. Like these are the different wells that the information were obtained from, and the plate details, and also the mechanism of action and the compound that they were treated with, and the concentration of compounds. And all the rows are the feature values that you would have extracted from the segmented objects. So this is how you load in the data and add all the annotations in. And now I'll quickly show why normalization is important and why we have to do that. Um, First thing, I will show making a similarity matrix, which is we can do it by going to Tools here and choosing Similarity Matrix. And since we were talking about using Pearson correlation, I'm going to be having the metric to be Pearson correlation. And you have two options to compute similarity matrix. One is on columns and the other one is on rows. I'm going to be doing it on columns now because we are interested in understanding the columns correspond to the different wells or the perturbations. So we're interested in understanding the associations between different perturbations. When you do it for rows, it is going to be saying the association or similarity between the different features, which we are right now not interested in. So I would choose columns and give OK. Okay, we have got the similarity matrix. But unlike the similarity matrix that I showed in the presentation, what we see is like an entire patch of red. What's going on? I would demonstrate why this is happening. I'll go back to the loaded data. The one tab that we have here is the similarity matrix, but I would go into the place where we have loaded the data. For example, here, if you, I will choose two wells corresponding to the DMOSO treatment, and also probably another well corresponding to another compound treatment. In this case, I'm choosing something where it was treated with microtubule destabilizers. And I'm choosing all the features that we have obtained from the different perturbations. Okay, I have missed choosing everything. Yeah. So once we have this, if I'm going to be going to the tools, I'm just going to make a chart to understand what's going on just with these three wells of data. So here in the chart type, if I'm going to be choosing column profile and the axis label to be, sorry, the color to be based on the well. So as you can see on the legend here, the column profiles are nothing but the features that you obtained from the three different wells, like from B2, C2, and E4. The B2 and C2 corresponds to the DMSO, and the E4 is the microtubule destabilizer that I've chosen. So you can see the features are not varying that much. Like everything is like overlapping onto each other. You don't see a much of difference between the DMOSO treatment or the microtubule destabilizer, but I would expect at least some phenotypic changes that should be going on behind. But it's not the case. Why? We will also have a look at the column scatter matrix to get an understanding of what's going on. Here, we are representing the same thing, but in a different chart form, which is the scatter matrix. And I would add some details about the labels of what these compounds are. And yeah, and also CP feature. So as you can see, this plot corresponds to the features that you obtained from the DMSO treated cells, and this plot corresponds to NucleoSO, which is the microtubule destabilizer. And we also have chose another DMSO well, right? And this plot corresponds to the other DMSO well. When you look at the plots here, you can see that when you hover over the feature, this corresponds to, you can see the CP feature name that's being displayed, which is cell shape area, and it is in the range of somewhere around 2000. And if you look at the data points way below, you could see the CP feature name. This corresponds to texture and also some of the neighbors. And these are in the range of like 50s, and the texture were like way below in 2. So you have values of different features ranging from somewhere from 2 to 2000. That is the same case with all of the features. So what we are right now seeing is there is a high like relationship between the number of the range of features that you obtained. When you go back to your plot here, generally in a heat map, what you would expect is like any blocks or squares that has red corresponds to higher values, and anything in blue would corresponds to a lower value, and anything in between would be having like a median value or a normal value. But in this case, I would just scroll up to show that's not the case. For example, if you see here, this feature corresponds to cell shape area, and if you hover over, it is in the range of 1300, and it is in blue. And if you go to, yeah, probably area shape and eccentricity, or this is having a value of 1 and it's red. But you would expect the opposite that's going on. Like any higher value should be in red, but any lower values in blue. That's not happening. Why is this is the case? Because what Morpheus does is it takes the range of the minimum and maximum of each feature. For example, if it is going to be area, it will take the minimum and maximum values of area and would make the colors corresponding to that. So each row here corresponds to different features, which is having a different scaling. Like some of them are in thousands and some of them are in ones or twos. If that is the case, it is not a very good way to compare between your different perturbations or cells. How do we bring in all this into a particular scale so that it is easier for comparison? That is where the normalization comes in, and that's why we have to normalize. So how this can be done in Morpheus is you could go over to the tools here and choose adjust. And you can choose robust z-score, which we generally use for normalization. And once this is done, you could now see, okay, why isn't happening once again? Okay, robust z-score. And oh yeah, I didn't click OK the previous time. Yes. Um, Now you could see, unlike the previous time where we were seeing the area to be in thousands, now we see somewhere in the range of 3. And whatever you look at, now you can see most of the values are within like probably -3 to +3 or in those ranges. And the lower values or the negative values corresponds to blue color, and the and like all of these uh, feature values have been brought into a similar scale. So now if we do a similarity matrix, or if we now do a chart to compare these, you can, I will choose a couple of cells like we did last time. Like, sorry, this is something that I don't want to do. Yeah. So I'm choosing a couple of cells of DMSO and probably microtubule destabilizer again, and all the features from these 3 wells. And if we now try to chart what's going on, like last time, when we look at the column profiles now, you see a lot of variation in the features that you obtained from 3 different wells, right? Like I will include the well details. So the legend now shows the B2 and C2 corresponds to the DMSO, and the E4 is the microtubule destabilizer. Now the column profile looks way different than what we saw before, and you'll be able to appreciate the differences between the perturbations now. So this is because we have normalized the data now, and that is why normalization is important before you actually start understanding or exploring the associations in your data. This is also can be more obvious when you do like a column scatter matrix, and I would label these plots with the compound name. Yeah, like this plot shows the features that you have obtained from DMSO, and this is in Nogresol. And this again, so when you look at, when you try to see the association between DMSO to DMSO, you see there is a high correlation between these 2 wells. But when you try to see the correlation between Nogresol treated wells to the DMSO treated wells, there's not much correlation, which you would expect because this would be bringing about some phenotypic changes in your cell. So this is like more obvious with the normalized data than the unnormalized one. And This makes the normalization step very much important. So now, having seen why normalization is important, now I would jump into showing you how to make a similarity matrix, which can be very well done here. Or before that, I would show the importance of feature selection and then go into the similarity matrix. So here, to know the importance of feature selection, I would do a similarity matrix based on rows this time, unlike the last time when we were trying to do it with on columns because we are trying to understand the associations between the different perturbations. Now we are trying to just see if there are similarity between the features that we have extracted from the different objects of interest. So when you do that, what you see here is like when you see like a whole patch of red, it means when you hover over it, you can see different values that you each square corresponds to, and and all of these values are the Pearson correlation coefficient because we have made a similarity matrix based on the Pearson correlation coefficient, and this means that the features uh, of like cell area shape of major axis length, ferrite diameter, maximum radius or mean radius, like all of them are very similar to each other when compared to the rest of the features. This makes sense because if a particular perturbation is changing your cell shape, you would expect the changes to be reflected in all of these because all of these features are very similar to each other, like the diameter or radius or ferrite diameter, like all of these are so similar to each other. So you can see that the area shape has been represented probably in multiple features. So when at the end, when you have all these features in, when you're making a similarity matrix, this would have like an additive effect when you're doing a correlation, which we actually don't want to be doing because this will not then actually reflect the biology that's going on behind, but rather all of these features would be dominating in there and would screw up or uh, would make the associations not the right way to be interpreted it. So what we would like to do is we would like to eliminate all the redundant features that you have in your data set. How you can do it? With Morpheus, we can actually manually curate it, which we generally don't recommend. In our typical workflow, what we generally do is we use a Python package called PyCytominer, which can be used to remove the redundant features and then understand the association. I've also linked a Jupyter notebook in the resources slide if you're interested in using the Python package. But Morpheus definitely gives a very good first pass if you're understanding the association that's going on and keeping in mind that these redundant features add to the associations as well. So this is why you need to make sure their redundant features are removed before you actually venture into exploring the associations. So having now looked into the importance of feature selection and also normalization, now we actually can jump into doing a similarity matrix and understanding the associations. For that, I would open another Morpheus tab, and here I would load in a data where it was already feature selected and also normalized. So I'm choosing a GCT file where it has already been, uh, where the pre-processing steps have already been done. So as I mentioned before, all of these rows corresponds to the feature values and the columns to different wells. So to make it more meaningful, I would add the annotations. So it was choose the options here and then go to annotations. And you can choose the different metadata that is associated with each of these wells. I would uncheck the ID because it is not adding much to the data. And then mechanism of action, plate information, and well information. So now we have all the metadata information that is being added and also the feature values. Now, before I actually make the similarity matrix, I would also color them. If you right click on one of the metadata, then you will have these options. Then you can choose show color. This will be helpful when later when you're seeing the associations, you'll understand it better why the colors matter. So this one, then you can choose show color for all the different tabs, which would be helpful to understand the association. So having done all this, then we can do a Pearson, like a similarity matrix on the feature selected and normalized data. Now you can go to tools and do a similarity matrix. So we are using Pearson correlation as the metric, and this time we're going to be doing it on columns because we want to understand the association between the different perturbations. Yes, now we can see the similarity matrix is looking something similar to what I showed in the presentation before. Um, so you can see like we have like big red patches and also different colors. And when you hover over it, all of the red patches corresponds to higher Pearson correlation coefficient, which means when you look at this point specifically, it is the D2 compared with the B2, as you can see it here. And this has a Pearson correlation coefficient of 0.9, which means the phenotype or the features that you extracted from D2 is highly similar to the one from B2, or the phenotype of the cells under these two different conditions are highly similar, which makes sense because both of these cells were treated with DMSO, and you would expect a high similarity between the replicates. Um, so uh, this is like just a part of the data set. You can have a look at the entire data by going to this magnifying glass icon here and fit to window. This is like the whole similarity matrix of the data that you have. And you would expect the diagonal to be in red block because you're comparing each well to the same well. So it would be the same values compared to each other. So you would have like a Pearson correlation coefficient of 1, and that you would expect to be in red. But the interesting part is anything beyond this diagonal line. Um, so before I do that, I would zoom in a bit so we understand what are the metadata that are associated with this and go in the details. So you can use the magnifying glass here and zoom in. Okay, now we understand what are the metadata associated with it. So the first way we can sort this data by clicking on them. Like if you click on the MOA and you see a tiny little arrow that is shown here, which means the data is now sorted based on MOA. I'm going to be sorting it based on MOA and the compound. To do two sortings, what you have to do is like click on MOA, you have like a tiny little arrow, and then hold shift and click on compound. Now you have like the data sorted based on MOA as well as the compound. You can also add another sort based on, again, holding shift and clicking on the concentration. So now your data is sorted based on MOA. Okay, before this, I forgot to do something. So before actually sorting the data, I would go in the options and link in the display option, you would link the rows and column. So when you sort them, both the rows and columns are not are in the same order. So once you have done that, now if you sort by just clicking on them, you see a tiny little arrow here, and then hold shift and also sort it based on compound and then based on concentration. So now what's going on is like you can see that all the actin disruptors, like all of this is where the color helps. Like this all have is all having the same color. So all the actin disruptors are kind of highly similar to each other than the cells that were perturbed with aurora kinase inhibitors. And you can also see that all the actin disruptors, although there are different compounds in the actin disruptors, such as the cytokeratin B, cytokeratin D, and latrunculin B, they all have like similar phenotypes when compared to the other compounds that the cells were treated with. Um, and as you can see, like when you hover over, and you can see that cytokeratin D is less similar to the latrunculin than the cytokeratin D, and so on and so forth. So this is how you can actually look at your whole similarity matrix and understand how correlated your different perturbations are. Rather than, I mean, apart from looking at the correlations, you also can look at the anti-correlations, because when you see like a big patch of blue, what this means is uh, the perturbations are like highly correlated. What do I mean by that? When you are treating the cells with cholesterol lowering drugs, this probably the phenotype or the features that you get from the cholesterol lowering drugs are very much different from what you're getting with aurora kinase inhibitors, and they are like highly anti-correlated. So when you see a huge block of blue, it means high anti-correlation, and huge block of red, it means correlation. So the interesting thing would be, like, rather than looking at the compounds that were treated with similar mechanism of action, if you see a correlation, I will just zoom out to understand that better. So rather than the whatever the diagonal, what you're looking at, if you're seeing some correlation between two different compounds, that means the phenotype, whatever is resulting from the perturbations from two different compounds, produce similar phenotypes. In this case, we are using compounds of treatment which we already know the mechanism of action. But if you have a compound, if where you don't know the mechanism of action, and if you include along with this data, then you would be able to associate based on your phenotypic data, whether it would belong to a certain mechanism of action class or not. So this is one way where you can sort and understand the association that is going on. And as you can see that, like the whole blue patch is corresponding to DMSO, and they are like in a separate block and from the ones that were treated with compounds. This clearly says that there are phenotypic differences between the control cells and the different compound treated cells. And we could also do a clustering here. Sorting is one way to understand the association. We can also do a hierarchical clustering, which I would show in a minute. But before that, you can also, when you click this back again, you zoom in. The sorting can be done like this is a forward sort, and this is a reverse sort, and this means this is unsorted. So that's just something to note. And we can now do like a hierarchical clustering, which can be done by going to the tools and choosing hierarchical clustering here. And the metric is going to be a pre-computed similarity matrix because we already have one, and we are going to be clustering both on rows and columns. With that, we will, yeah. So now this is all clustered. If I fit this into window, now you can see like the whole DMSO is under one cluster, and the rest of the uh, compounds based upon their cellular phenotype, they are clustered under different clusters, and where you can understand the association that's going on. This is one way of clustering that you can do in Morpheus. There's another way of looking at an unsupervised clustering, which is like doing a Disney plot. So which can be done by going to the Tools menu and choose a Disney plot. And we can do the Disney plot here. It is taking a while. Okay, it is taking longer than I expected. Wait. Yes, now we have the Disney embeddings. How we can visualize this is we can go to the tools again and choose chart. And based upon the embedding that we would have obtained, we can go to the embedding and choose the two different things that would have resulted from the Disney, which is T1 and T2. And I'll increase the symbol size to something 5 so that we can have, yeah, better visualization. I'm going to be coloring those data points based on probably mechanism of action of class. So this is another way of looking at your clusters. As you can see, like since we colored the different classes, we can see the blue class corresponds to the DMSO, and all of them are clustered separately, and all the other MOAs which belongs to a different class are clustered. So they are clustered based on the different mechanism of action. So this means that the cellular phenotype associated with these perturbations are more similar to each other than the DMSO, and so on and so forth. We can also try coloring them based on compounds. And yeah, and when you try to maybe try it based on wells, they would be totally completely different because we shouldn't expect a grouping based on wells. So this is another way of doing a clustering with the microscope, with the data that you would obtain from your microscopy images. So these are the different ways that you can look at the associations using Morpheus. And now, having seen all the associations, if you're interested in, okay, I do see these associations, but how do I know which features have actually driven these associations or dissimilarities? To have a look at that, what we can do is we can go back to our data that we loaded in and go to Tools and do a marker selection. What do I mean by marker selection is we are trying to understand what are the different features that have been altered between your associations or dissociations in the data. So we can choose a metric of t-test. And the field that I want to study is based on probably MOA, or the mechanism of action. And I'm going to compare the control with other class of compounds. So I've chosen class A to be DMSO, and the class B probably cholesterol lowering drugs or DNA damage, for example. Yes. And now if we do a marker selection, what the Morpheus have done, it has chosen all the wells that were treated with DMSO and also all the wells that were treated with the DNA damage. Here you see only the DMSO, but if you like scroll over, you would see the DNA damage as well. So on the rows, you have all the features that differentiates these two classes. We will sort by p-value. And these are the top features that drives the differences are the associations or like the dissimilarities between the data. So since we have chosen the control versus the DNA damage, you would expect more features from nuclei. And of course, we see more features from nuclei, the area, shape, and textures. But we also see some features related to actin. Mm, which again, like these are the interesting things that you would find. Like, although you see the associations, there are like features which would have actually driven these association, and you can understand those features using this marker selection feature. And yeah, like this is one way where you can know the features that drive the association that is going on. And you can also do a quality control with the data that you have. How you can do that is generally the Morpheus does a relative color scheme. So when you go into these options, the quality control is more to understand if there are outliers in your data or not. Uh, to do that, we can uncheck the relative color scheme, which Morpheus generally defaults to. You can put something, the minimum value to be like 100 and also the maximum to be minimum to be -100 and maximum to be somewhere in 100. And yeah, those probably fit this. Sorry, rather than seeing a subset, I would like to fit it to a window. Now, when you, so all these blank white regions are something values in the normal range, but there, you can still see there are something highlighted in red. Probably I'll zoom in because it is not super obvious. So, yeah, like you have something in red or okay, I am still trying to get like, yeah, here. So the rest of the regions are in the similar scale, but these are kind of highlighted or having a brighter color because these are having a higher value than rest of the data. And like anything in the range of -6 is something, oh, fine. But when you have something in the range of like hundreds or thousands, then you need to make sure you understand that there's an outlier in your data. And when you're exploring the association, you need to make sure that you take that into account when you're trying to interpret the biology. So this is one way where how you can check if there are outliers in your data or not. So yeah, these are some of the things that I would wanted to show in the demonstration. So far, I've been working with the GCT file. You can also do this with the CSV file. I'll also show how to do that so that then we can get to the hands-on exercise part. So if you load in Morpheus and choose a file, so far I was showing you the GCT file format. I've also provided the CSV and the GCT file format in the materials, tutorial materials. So if you choose the CSV format, the only extra thing that you need to do is what it generally, when you open the data in CSV format, we have all the metadata information before this, and it asks us to choose the table cell that contains the first data row or column. So the first data row or column would be the features that is cell area shape, and anything before that are just the metadata information, like the plate information or which well it came from, and so on. So we want only the data to be loaded in the similarity matrix. So you choose some column like these, and you have all the metadata towards the left. And when you choose that,
Deep data.
the cell.
The one below.
Oh, okay. Yeah, perfect. Yeah, rather than choosing the column name, it has to be the actual data. Yeah, this one. Thanks for pointing it out, Esteban. And yeah, once you've chosen that, click OK. And now you see the rows are the information from different wells, and the columns are the features that you obtained. So the only thing that you need to change before you jump into the similarity matrices with the CSV files are you have to make a transpose, which can be done like tools and transpose. And now this looks more similar to the one that I've shown before with the GCT. So yeah, these are the different things I wanted to show. And with this, I will stop sharing my screen. If there are any questions, I'm happy to answer. Otherwise, the materials of the data that the GCT and the CSV files are provided in the um, link to the tutorial materials. And these were the different things which I wanted to show. And of course, I probably had all those. I would give some information on the data that we have provided. Um, we have provided a data set which is images from MCF7 cancer cells, which were treated with 113 different compounds at 8 different concentrations. And they were imaged for DNA, actin, and tubulin. And the negative control is DMSO. And all the features are in the GCT file as well as the CSV file that we have provided. And you also have a PDF, written PDF in the tutorial material, which you can go through and follow accordingly and try the data on the Morpheus tool. And if you have any questions, please put it in the chat. I'm happy to answer. And the TAs will be helping you guys too. Thank you. And I would like to acknowledge the whole Simile team. And as well as I would, again, like to highlight the fact it would be very helpful if you could fill up the image analysis survey. And thanks, Copa, for the fund. Thank you.
Just a final plug. We also sorry, just a final plug before we separate. We also added link to a I2K post-survey event. So this is to give feedback on this whole set of tutorials that have been going on in the past few days. It will be also very useful if you can complete that. I will ping it again in the resources. Thank you.
Since we are like very few members, I think we can just do it in the same room. So one of the participants can volunteer to share the screen, or others can help in reading the PDF and going over the Morpheus, whichever way it works. Or if you want to do it on your own pace, feel free to do that too. If you have any questions, post it. But we highly encourage sharing the screen. And also feel free to unmute yourself if you have any questions. I hope you were able to download the materials and you were able to get the PDF. Give a thumbs up if you were able to do it, even if you don't want to unmute or share your screen. I do see a question. Can the Morpheus tool be used for statistical analysis of non-microscopy data? Thanks for the question, Tyagarajan. Do you mean by non-microscopy data, is it like a genomics or proteomics data? Can you provide a bit more details on that, please? You can also feel free to unmute yourself and ask questions too, or put it in the chat, whichever is comfortable for you. I assume you're typing more details, but yeah, I'll wait for you to answer. Any kind of generic data in machine learning. So you mean like any numbers? So like it can be any numbers, irrespective of whether it is image-based profiles or transcriptomics or genomics, but you just need to understand what the numbers are. And if you have enough, yeah, it can be used in Morpheus. And of course, the statistical analysis, probably what you say is Disney or something like that, or t-test, or yeah, it can be definitely used. If you have an file with full of table full of numbers, yes, it can be definitely loaded in with the CSV or the GCD formats. For feature association, yes, yes, you can definitely do that. Yeah.
Yeah, to build on a little bit on that answer, the data that we've seen in this exercise was the features were extracted with Cell Profiler, but you can also extract features using some sort of deep learning model in which the features are non-interpretable, but you can still group them and do the matrix association in Morpheus.
You asked what is like GCT format? It is gene cluster text, that is expansion. It is nothing but a similar a table like how you get your CSV format, but it is just another format which is more specific for the gene expression data, and it's called as expansion. It's like gene cluster text. You can obviously convert the CSV to GCT if you're good at like Python or something like that. You can probably there might be tools just to convert CSV to GCT, and you can also definitely use the CSV as we have pointed out. That's possible too.
Depending on
Thanks for the thumbs up. Okay, I do see another question. Really, thank you for this. I know there's a lot of time, but we'll email if I have questions going through the exercise. You still have time till 12:30, but yeah, definitely at your own pace. If you want to do it at your own pace, feel free to post it on the forum, or yeah, that would be a very good way to have an interaction so others can also go through your questions. If you have a similar question. But thanks a lot for joining. I hope you were able to download the materials and are working on the data, and feel free to drop in questions if you have any at any point of the time doing the exercise. And if you want to like understand more about the PyCytometer, which I mentioned during the talk, if you're well versed in like if you need not be well versed, if you're kind of comfortable using the Jupyter notebooks, and if you want your data not just being a first pass with Morpheus, but you want some more information from it and make it more uh, friendly or more appropriate with the data associations, I would recommend watching videos on PyCytometer, which is on the YouTube. Again, the links to the YouTube are provided in the resources slide in the slide deck. Feel free to check that out if you want to use the PyCytometer package. Hello. And the Jupyter notebook that we have linked also has like every steps in there in all the cells. You just need to provide your data, and it run all the PyCytometer steps. So feel free to check that out too if you're interested in using the PyCytometer package. Then does Morpheus support only PCC or other kind of association metrics too? That's a good question. We generally start with Pearson correlation coefficient, but I would definitely look at it right now to see. You do have Spearman rank correlation, Kendall's correlation, cosine similarity, Jacquard distance, and Euclidean distance. These are the metrics that you can do the similarity matrices on. Thanks for the question. Okay, I see another question. I worked on co-localization analysis where PCC is also used as a measure of association of per pixel intensity. Yes, like you have Pearson correlation coefficient, Manders correlation, Spearman too, yeah. I'm assuming then you're also working with a lot of image data and you're trying to do machine learning on your image data. Thanks. Okay, good luck.
Yes, it is the same Pearson correlation coefficient. It's just used for different values. In one case, you are doing the pixel value in one channel versus the other, whereas here we are doing a feature in one group or one well in this case versus another well. But it's the same math behind it.